We're very excited, now that we know that BTK inhibitors have a very important place in the treatment of Waldenström's, and more importantly, that this is supported by the underlying genomics, knowing that, the mutated MYD88 gene can actually transactivate through its protein production. Bruton's tyrosine kinase makes this spot on when one thinks about, the disease itself. So being able to optimize now BTK inhibitors is really the challenge. And there are a number of areas which we're very interested in pursuing. One is the combination with Cxcr4 inhibitors. We're currently looking at the ulocuplumab monoclonal antibody that binds the Cxcr4 receptor and prevents its ligand SDF1 or CXCL12 from engaging it. This is actually a very important trial because this is the way we hope to optimize the use of BTK inhibitors in the Cxcr4 mutated Waldenström patients. And that's about 40% of all patients. So it's a very, very large number, particularly those patients that carry the nonsense mutations where the drug shows lower responses, deep responses, and shows a longer time to attaining a major response. So the use of Cxcr4 antagonists represents a very nice and innovative way to be able to optimize BTK therapy in the patients that are Cxcr4 mutated. The other is targeting BCL2. We were among the first to publish data that actually looked at the synergy that can be conferred when you use a drug like venetoclax that targets BCL2 in combination with ibrutinib. And this is actually a very important point because venetoclax is very active in the treatment of Waldenström's disease. We know from the data that we presented at EHA that patients, regardless of prior exposure to ibrutinib or their Cxcr4 mutation status, can attain responses to venetoclax. So being able to combine venetoclax with ibrutinib is also a very important step forward, and a trial looking at this combination is planned for the first quarter of 2019.
I think a really interesting question for BTK inhibition in CLL is whether the addition of an anti-CD20 monoclonal antibody adds efficacy, either rituximab or obinutuzumab. There was just an FDA approval for ibrutinib combined with obinutuzumab in frontline CLL just last week. Looking at data closely across trials, I don't think it's not obvious to me at least that the anti-CD20 therapy, in this case the obinutuzumab, is really adding to the efficacy of what you can get with ibrutinib alone. So I think we need additional studies to help us tease out whether you need to add anti-CD20 therapy to your BTK inhibitor in CLL. I think that's an unknown right now. As far as other rational combinations, I think one of the most interesting combinations right now is the idea of combining a BTK inhibitor with venetoclax. Venetoclax is a oral small molecule BCL2 inhibitor. It has an approval now in CLL. It's very active in CLL. It's active in other lymphoid malignancies. It's quite active in mantle cell lymphoma, and it just received approval in elderly AML. It has activity across a variety of hematologic malignancies. We know that if you can knock BCL2 down inside cancer cells, those cells just become easier to kill. So venetoclax is a really attractive drug for combination therapies. And there are now published reports of the combination of venetoclax and ibrutinib in relapsed mantle cell, for example, and we see very high complete response rates, much higher than you would see with either agent alone. And I think you're going to start to see a lot of combination data in CLL. Some of this has been presented at meetings, BTK inhibitors like ibrutinib in combination with venetoclax. We don't have anything yet in publication, but over the next year to 18 months, I think we'll see a lot of data combining BTK inhibitors with BCL2 inhibitors. And the real goal here is to come up with strategies that allow us to give patients time-limited therapies. It's one of the less desirable traits of BTK inhibitors is that patients are required to be on therapy chronically. And that can be years and years and years. We would love it if we had therapies that patients could take for a shorter duration, 6 months or 12 months or even 24 months, but then stop, so that patients can enjoy nice, long treatment-free intervals, off treatment, free of all side effects of treatment. That's really the next frontier in CLL therapy, I think, is you're going to see a lot of combination studies with a goal of establishing the efficacy of time-limited therapies so patients can get therapeutic breaks, which would be good for quality of life reasons.